Hello and welcome. Today we're going to have a discussion around thermal stability data. My colleague and I will discuss various types of biologics, what their nanodifferential scanning fluorimetry data looks like, and share some tips and tricks along the way. So I'm Dr. Stefanie Kall, and as part of my job on the product squad, I speak to a lot of researchers working in the biologics field. Over the years, I've learned a lot about your most common challenges in the lab. And in previous webinars, I've covered much about the different experiments possible for getting stability information about your biologics. But today I wanted to take that approach a little differently. Let's look at some real life examples of nanoDSF data and discuss not only what you're trying to assess with each type of biologic, but also dissect the data to help you understand what specifics to look for and kind of like what variables to design your experiments around. To help me out with this, I have brought in my colleague, Dr. Bridget Milorey. She's an applications specialist, and she has spent a lot of time in the field helping researchers use nanoDSF and other stability assessment technology in order to make decisions about therapeutically relevant proteins. Hey, Bridget.
Hi, Stephanie. Thank you very much for the introduction, and I'm really excited to share what I have learned about data analysis and experimental design throughout the years.
I'm super happy to have you. So before we get into specific experiments, I do want to briefly review how nanoDSF data is generated. There's a much longer video that deep dives into this topic titled Thermal Stability Measurements for Biologics Developability Profiling. If you want to know more, click the link below. But for now, I'd just like to cover the basics so we're all on the same page. So nanoDSF uses the intrinsic fluorescence of a protein to assess its conformational stability. As proteins unfold due to chemical or thermal denaturation, their tryptophan and tyrosine residues get exposed to solution. So what happens is this changes their emission maximum of the protein. And we follow that emission very precisely at 330 and 350 nanometers. And with that information, we see how the fluorescence changes in response to these thermal denaturation gradients. And specifically, the point during a thermal denaturation ramp at which 50% of the protein is folded and 50% is unfolded is the melting temperature, or TM. Ranking TMs is a super common method for finding the most thermostable biologics candidates or stabilizing formulations conditions that are worth developing or characterizing further. I think it's also worth noting that sometimes it's easier to find that inflection by looking at the first derivative profile, especially when you have multiple domains unfolding, which reveal multiple TMs. This is an example of what that looks like. But now I want to turn to Bridget. Bridget, would you do me a favor and walk through sort of the details of what a monoclonal antibody's nanoDSF profile looks like, just broadly and in general?
Yes, definitely can. So we'll pull up a slide here, and what we're looking at actually here is the NIST monoclonal antibody, or the NIST mAb. And you can see that in the nanoDSF unfolding profile, there are multiple TMs, which is typical for mAbs. And as Stephanie just said, these TMs correspond to different domains or multiple domains unfolding. This data set here has three domains, or shows three domains unfolding, and in mAbs, those would be the FC1 domain, corresponding here to TM1, the Fab domain with TM2, and the FC2 domain with TM3. I want to point out that you won't always see three unfolding events or three TMs, because sometimes the Fab and the FC2 domains actually have very similar melting temperatures, and you'll lose some resolution there. And we also see the first derivative view here, which I agree with Stephanie, this is often easier to find the inflection point. And as you can see here, the markers line up with the peaks in the first derivative views.
Right. So thanks for explaining the data output with this kind of model antibody. Can you briefly talk about that black marker labeled T-on?
Yes, very good to point out T-on. So T-onset is an additional parameter that we can use to evaluate the sharpness of inflection. So this is another useful indicator of how stable a protein is. Generally, a higher T-on or higher onset of melting is indicating a more stable protein. But you can also look at how close in temperature T-on is to Tm to see if you have a sharp transition, which would indicate a more stable protein than something that starts to unfold slowly early on in the temperature gradient.
Yeah, that makes sense. Something's less stable if it takes sort of a while to unravel. So, is there anything else we should think about when we just look at this kind of data?
Yes. So this data here, as I indicated, is the NIST MAB. It is in its histidine buffer, which is a very stable system. Not every antibody that you are looking at will be that stable. And I also want to point out that nanoDSF is really just one piece of a puzzle. If you want a full stability picture, there are combinatorial approaches that become necessary. So we have actually provided some links to other videos that take a deeper dive into that concept.
Yeah, so thanks, Bridget. So as we can all see, looking just at a single experiment for a super optimal monoclonal antibody in its ideal buffer, there's a ton of information to dissect just from the nanoDSF profile. So before we start talking about these specific experiments and all the data, Bridget, can you give us some insight on what is good advice for just setting up a nanoDSF experiment in general?
Of course. So keep in mind that a lot of the aspects of sample handling that I will describe are going to be specific to our Prometheus Panta. So there may be some other considerations to take if you are using a different approach to collecting TMs. But generally speaking, replicates are going to be best practice. However, early in development, some people are very limited to sample quantity. So in order to maximize throughput, you may do single runs of your samples. We do our experiments in capillaries. So it's always important to consider handling the capillaries properly. You do not want to touch the capillary in an area where the optics are actually reading a measurement. And also for nanoDSF being an intrinsic technique, you want your sample to be about 80% pure minimum. So this means that your samples will require some light cleanup after lysing. And also keep in mind that any other protein, such as a chaperone, will also have an unfolding shift in nanoDSF. And then one last tip that I have, I like to spin my samples in a refrigerated centrifuge before running them.
Oh yeah, that's actually a really interesting point because I don't always see researchers spin their samples. Can you talk about why you kind of recommend that?
Yeah, so with your sample, if there is any dust, dirt, or other debris, or if there are already aggregates present, this is just going to lead to noisier data compared to if you have a pure protein sample. Another thing to consider is this will also prevent or help to prevent the formation of air bubbles, and that those would interrupt your acquisitions.
Yeah, that's actually another really good point. A lot of researchers really want to actually know or see the aggregation in their samples. So can nanoDSF help with that work?
nanoDSF alone won't tell you anything about aggregation. However, there are other ways to look for aggregation in parallel. For example, with the Prometheus Panta, we have back reflection optics and dynamic light scattering to help follow the colloidal stability over your temperature ramp. So with that back reflection optic, you can detect the presence of large amorphous aggregates. And with the light scattering measurements, you can also see smaller size changes that are associated with partial unfolding and oligomerization.
Okay, cool. Yeah, thanks. And then finally, I just want to ask, we get a lot of questions about how low of a concentration you can do with these experiments. So can you just speak to that a little bit?
Yes. So while you can push the lower limit of detection for nanoDSF down to about 5 micrograms per milliliter, it's really best to use at least half a milligram per milliliter, especially if you're in the triaging phase. And this is to ensure that you have a good signal to noise resolution on your unfolding data. And this way you can feel really confident about the differences that you are detecting between candidates.
Right, thanks. And we're going to see some of just why having this high resolution data is really valuable very soon. And so, yeah, now that we've talked about these basic guidelines for designing a nanoDSF experiment, I want to start talking about specific examples and how to really apply this to different aspects of biologics development. So first, we have monoclonal antibodies, which I'm sure many of you realize are a huge market. They represent over $200 billion worth of the biologics market, and they're continuing to grow at a rate of about 11% per year. So I want to start by talking just in general about antibody engineering, particularly monoclonal antibodies. Some of you may start your biologics workflow with a known antibody that you want to improve in some way, for example, by increasing its affinity for its antigen or improving its developability characteristics so it behaves better once it reaches the clinic. Others may be starting completely from scratch, having chosen a new target and having to pull out a promising monoclonal antibody candidate from a library. So, but either way, no matter what you're doing, you have an antibody and you want to improve it. And making rational mutations to that antibody is a time-tested method for tweaking its developability parameters. And modern machine learning is also being applied to boost that process. However, these machine learning models need really high quality data in order to predict which mutations you make are going to increase thermal stability. So enter nanoDSF. Just pull up a slide. So here we're looking at a few different constructs that only vary by a few amino acid substitutions. You may recognize this data if you've watched Explore Prometheus Panta, Biologics Developability Assessment with 4 integrated technologies. My colleague Nathan talked about the data in depth there, including the other stability data measured in the same experiment, which Bridget sort of mentioned earlier. But right now, I'm just going to have her really walk us through the unfolding curves here. So, Bridget?
Yeah, so first of all, let's take a look at the figure as a whole. So I want to point out that you have this originator or parent mAb that is appearing as the purple data set. So if you look to the unfolding profile on the top panel, this is the profile that has the highest TM value. And you can see there are subtle shifts in TM when you're looking at these teal data sets, which, as you can see, have 2 to 3 point mutations in the construct when compared to the originator map. And what I want to point out here, sorry, what I want to point out here,
sorry, I was going to follow up, but yeah, what do you want to point
out? That you not only see the shift in the TM just from these 2 to 3 point mutations, but you can also see a pretty big impact on the overall profile shape.
Yeah, I was going to say, even when you were just talking about the TMs, but it's really interesting how much they change with just a few point mutations, especially when you think about how large a monoclonal antibody is. So can you really briefly talk about what the experimental setup was here?
Yes, so for the experiment, the concentrations of these samples was 2 mg per mil, and the researchers did single runs for their measurements. And this was because they really felt confident enough in the data resolution with a single run in order to accurately compare their constructs to the originator.
Awesome. And then is there anything else to think about if anyone watching wanted to set up their own experiment like this?
Yeah, so for an experiment like this, where you are comparing to a reference, it is very critical to run all of your samples at the same concentration and all in the same buffer conditions.
Yeah, those careful controls are really critical just to make sure that those differences in constructs are related only to the sequence change and not any other sort of variable like concentration or buffer. So let's talk about next antibody drug conjugates. So ADCs are a really cool approach to targeted medicine. They take a small molecule that is optimized to target a disease-causing protein in the body. They fuse it to the antibody and it gets that cellular level targeting of a mAb. This helps reduce the off-target effects from the small molecule. They are a growing proportion of the overall biologics market. They're currently worth about $7.8 billion, and they're expanding at about 11% year over year. A lot of these ADCs are specifically used to target cancers because they can really hone in on those cancer-causing cells while ignoring the good cells in your body. So now, of course, when we think about the experiment, the conjugation process can destabilize the antibody, whether from the harsh chemicals that you need for the conjugation or just from the act of like bringing that small molecule in to perturb the antibody's environment. So Bridget, I'd like to hand things back over to you to walk us through this data example.
Yes. So again, let's take a look at this figure as a whole. So we have here two unfolding profiles. One is for the simple naked mAb, and the other one is for the ADC, or that antibody drug conjugate. And with really high resolution data like you see here, you can identify exactly which domain on the antibody got conjugated by that drug. And we see this in the rather dramatic shift in TM1 for that FC1 domain. And you can kind of compare that to the shift that you're seeing for the Fab/FC2 domain. Now, tying this back into something we said earlier, this here would be a good example of an unfolding profile where you actually only see the two TMs or two transitions. So we lost resolution between that Fab and FC2. I also want to point out that you can see some peak broadening if you look to the first derivative view for that FC1 domain. And this suggests to me that there is some destabilization of that domain coming from the conjugation process, or maybe even just the change in environment due to the nearby drug. However, I will say that the similar intensity that we are seeing is indicating that the conjugation process did not actually damage the mAb, which is something very important to consider.
Yeah, it's actually really neat to see how just like adding that small molecule to the whole complex protein biologic really alters its behavior. Can you talk a little bit about what aspects of ADC development are going to have an impact on the final stability profile of the ADC?
Yes, definitely. So a couple things to consider here that can impact the ADC. First, you have your conjugation process. This is something that often requires harsh chemicals, so it can be worth the time to investigate if you can reduce the amount of active reagents, and that will reduce stress that you're putting onto the mAb. Another aspect of the conjugation process that you can consider is the length of incubation time. That can also have an impact on the mAb. A couple other things. So the drug, some things to consider regarding that. Length of the linker from the antibody to the drug, and also the solubility of the drug plus linker. And then one final thing to note, you are still looking at this antibody, even though there's a drug conjugated to it. So you still want to consider your buffer conditions and look for optimum buffer components.
Yeah, so thanks. Like ADCs are a really cool newer tool for being really specific about disease targeting, but their complexity definitely makes them a little more challenging to develop. So thanks for sharing with us some good approaches for that work. So next up, I do want to talk about biosimilars. The goal of a biosimilar is to take a marketed, well-characterized therapeutic antibody and develop a new antibody-based treatment that is close enough to the original that regulatory bodies will allow it to reach market with a reduced amount of R&D, since the original mAb was already deemed safe and effective for treatment. The biosimilars market is growing even faster than ADCs, especially with a lot of these original patents starting to dry up. And it's currently worth about $30 billion. And it's growing fastest among submarkets at about 17% year over year. Biosimilars offer opportunities for companies that weren't first to market to compete against the original patented therapy, potentially driving down drug prices. They also offer an avenue for other countries to manufacture drugs more locally. Biosimilars exist for many treatments, although monoclonal antibodies that target autoimmune diseases are amongst the most common. So the goal of biosimilar development is to make a product that is as similar as possible to the originator mAb. Bridget, we have another case study. Will you walk us through biosimilar development?
Yes, absolutely. So turning to the slide here, this data comes from a group at UGA Biopharma. And what they decided to do in their approach is to look at the unfolding profile as a whole. So they actually created their own algorithm for determining how much variation they saw in unfolding profiles when comparing to their reference candidate.
Yeah, so the algorithm then tells them how much their biosimilar candidates deviate from the overall profile of the originator. That does seem like a pretty useful trick for ranking the best candidates.
Yes, it is definitely a very useful trick. And it also shows that it's really critical to have that high resolution data here. And for experimental design, in order to increase the resolution, what I would suggest is running a slower temperature ramp rate and using relatively high protein concentrations to see higher signal to noise, and so that you can really detect the differences in these profiles. One other thing is that using the parameters Tm and Ton alone don't really give you the whole picture. So that's why you need the high resolution fingerprint, as I will call it, from nanoDSF.
Yeah, that high quality data output seems particularly critical for this kind of work and especially developing this algorithm. So can you share any other just experimental design type considerations here?
Yes, so as before, ensuring the same concentration of sample and same buffer for everything, that's going to be critical. And I will also point out that in the figure that we're looking at on the slide, this has an example for the best comparison and the worst comparison, but you can actually see the full study and look at all of the comparisons. We have a link to the app note, and that is going to be titled Rapid and Precise Biosimilar Candidate Profiling by nanoDSF.
Yeah, thanks again. So as you can see, the general considerations for experimental setup are really similar for a lot of these experiments. But what you're really trying to understand from the data is just like what you're trying to pull out of that information is a little bit different depending on what kind of type of biologic you're developing. Yeah, so last I want to talk about formulation. This is work to optimize your buffer for long-term storage. You want a therapeutic that you spent all your time optimizing and characterizing to remain folded, active, and unaggregated on its way to the clinic. This is super critical for efficacy and safety. Formulation isn't a dedicated market per se, but any complex therapeutic you develop will require at least an assessment of long-term stability. But that means that even if your group isn't personally responsible for the buffer optimization work, it is something that will ultimately impact the stability of the biologic you're developing, and someone down the line has to do this work. Now, the amount of flexibility you have with your buffers may vary. Buffers have to meet certain safety requirements for administration, and it's not always possible to perhaps store your antibody in one buffer and change it out into an administration buffer. So you lose a little flexibility there. But there are a lot of regulatory agency approved buffers already sold for this type of screening work. So I'm going to turn it back over to Bridget. She's going to walk us through an example with Herceptin and a few different buffers. So Bridget, can you just tell us a little bit about the data we see here?
Yes, so as you pointed out, this is three unfolding profiles of Herceptin in three different buffer conditions. Here we're just looking at the first derivative view for ease of explanation. And the biggest takeaway here is that the PBS buffer at pH 7.4 is the best. When you compare this to the sodium acetate buffers at pH 4.0, we can really see how that pH change has destabilized the first unfolding, and you can see that in the shift of TM1. We're also looking, if you just focus on the sodium acetate buffers, we're just looking here at an effect of adding even more salt. So with the additional salt, you see another destabilizing effect.
Yeah, so that's interesting because here it would seem that a higher pH buffer is more stabilizing, but is this generally the case for antibodies?
No, actually, it's not. And in fact, this is a great example of where those combinatorial approaches are really critical. So, lower pH does tend to destabilize that FC1 domain. We can see that again in that TM1 shift. But it typically enhances the colloidal stability. And we're not going to get into the differences here and that combinatorial workflow, but it really is worth noting that a pH of less than 7 tends to decrease your overall propensity for aggregation.
Yeah, and I've definitely talked about this before in previous webinars, but the power of multi-parameter assessment is really striking when it comes to formulation work like this. So are there any other experimental setup considerations here?
So, in the case of buffer screens, you may be screening dozens or hundreds of buffer conditions. So, it's again critical to keep things very consistent, concentrations of proteins and things like that. And in this case, liquid handling can really provide a big help for the experimental setup. And I will also say that if you are working from a stock and diluting into a new buffer, you should at least try to do a tenfold dilution so that there's only about 10% of that old buffer present. And again, thinking about buffer exchanging, dialysis or buffer exchange columns are going to be the best practice. But if you are limited by the amount of material, consider that these steps will require you to recheck your protein concentration afterwards.
Yeah, and as usual, it just sounds like it's all about careful planning for whatever type of experiment you're running, and in particular for formulation. So, can you just share a couple or whatever components people might screen in their formulation work?
Yes, so pH, salt type, and concentration, the presence or absence of detergents and adjuvants are all common things to screen for in a buffer composition.
Right. So thanks again for helping us break down all the data from these experiments, Bridget. So to wrap up, I'm going to ask you a few rapid fire experimental questions. It looks like we're coming to the half hour. So feel free to stick around while we ask some of these Q&As, and then we'll get to the questions that have been coming in during the event as well. But these questions I'm talking about come from previous question and answer sessions that we didn't have time to cover, but that I thought were really helpful to talk about when we think about experimental design. So the first question is, does nanoDSF only work for proteins or biologics? What if I have nucleic acids or small molecules?
So generally speaking, nanoDSF is only for proteins, and this is because it uses the intrinsic fluorescence coming from tryptophan or tyrosine residues. However, if you're trying to look at protein DNA or protein RNA interactions, nanoDSF can still be a useful tool for you. So this is because you won't optically see the nucleic acids, but if they do bind and stabilize or even destabilize your protein, you will be able to see that effect in the unfolding profile. And it is worth noting here, because of the nature of the question, if your goal is to look at the unfolding or denaturing of a nucleic acid-based system, there are other DSF techniques that use extrinsic dyes that could be useful.
Yeah, and in that case, is there anything to consider about nucleic acids when you're adding them to your protein-based experiment?
Yeah, definitely. So nucleic acids tend to be highly positively charged because of the backbone. So that may have an impact in your formulation.
Right, of course. And then what about the small molecules?
Right. So the small molecules will also not be seen optically in nanoDSF, but you can still do a thermal shift assay to quickly assess whether there was a binding event. Um, Generally, small molecules tend to stabilize proteins that they bind to. And here we have a nice example of that on the slide. And what we're looking at here are the thermal unfolding profiles for BSA when in complex with increasing amounts of ibuprofen. And you can see that as we have increased that ibuprofen concentration, you see higher TM, so a positive TM shift. That's indicating we have stabilized the BSA. And again, while the ibuprofen as a small molecule is not seen by the optics, what we can see is that stabilizing effect.
Yeah, thanks. So TSAs are definitely a really cool, helpful tool to use, nanoDSF, in order to do your small molecule screening. So then my next question is about throughput. What is the throughput of nanoDSF?
Throughput is going to vary by the instrument setup that you have. So I will explain a couple different options. With the Prometheus Panta, if you prefer to load individual capillaries, you can load anywhere from 1 to 48 single capillaries in a single run. We also have these capillary chips. Those are 24 capillaries that are pre-mounted on a scaffold, and they're spaced so that they fit right into a 384 well plate, making your sample loading fast and easy. We also have an option for walkaway automation, and this would allow you to run 1536 single point measurements in just a couple clicks. And there are other methods for TM determination, which Stefanie has actually talked about in an earlier video. So those might have different throughput options.
Yeah, and I do want to emphasize something else I talked about in that video, which is that TMs are a really popular screening parameter, specifically because it is fast and easy to set up a lot of experiments at once, especially if you have liquid handling at your disposal. And that's part of the power of screening TMs specifically. So speaking of that screening, what are the benefits of nanoDSF over something like extrinsic DSF with Cipra orange dye?
NanoDSF is going to have generally a lower limit of detection, which is particularly helpful if you are sample limited. NanoDSF also has greater buffer flexibility. So for example, with Cipra, it typically does not do well when detergents are present. Another thing I would consider is that the addition of a dye can be falsely stabilizing. And then also with nanoDSF, you can run this in parallel with other technologies, which again, we're not focusing on that today, but that would give you a lot of extra parameters to consider.
Awesome. And just in the interest of being fair, I do want to pull up here a little bit about how DSF works, which it uses the extrinsic dye, which basically lights up as the protein unfolds. But can you talk briefly on what other advantages there might be using extrinsic DSF?
Yes, so I like to be fair too. So it is worth noting that some unfolding experiments with Cipra would typically require a more common or a more typical spectrophotometer. And they tend to have pretty decent throughput. So most are using at least 96 well plate format, and they can be relatively inexpensive.
Yeah, thanks. Again, I did talk about that more in another video if you really want to deep dive into the topic. So my next question is a little bit theoretical, and it's specifically about the characterization stage of a biologic. This is kind of directly quoted from the original question. Doesn't the TM just need to be higher than the room temp or the maximum temperature it will reach during transport or storage?
Yes, so I really like this question. It is true that if a protein's TM is any lower than those temperatures for storage or transport, then it is definitely, without a doubt, not a good candidate. However, when we're thinking about screening, a higher TM is going to indicate a more thermostable candidate. And when you're screening for a lot of stuff at once, you can imagine that even if you're not going to take your protein and walk into the sauna or go sit in a hot tub, you're still just trying to find a candidate that gives you the highest likelihood for success long term, so in the clinic. And being thermostable is just a nudge in that direction.
Yeah, thanks. So along that line, researchers have to find their best candidate. So they're likely looking at more than just TM or even TM and TON. And we have run into this kind of combinatorial analysis idea several times today. And our last question, I just want to expound a little more on that, which is, how does thermostability correlate with other stability parameters?
Yeah, so that's a great question too. And let me just use the rest of the data from that point mutation analysis we were talking about earlier, and this will help me illustrate the importance of assessing other parameters as well. So here on the slide, we have a side-by-side view comparing the colloidal stability versus the thermal stability. And what I'll point out is that generally a less thermal stable candidate is more likely to have an increased propensity for aggregation or measured turbidity, though it's not really guaranteed. And this is where it becomes really critical to evaluate a full matrix of parameters.
Yeah, I mean, I definitely talk ad nauseum about the importance of this multi-parameter characterization. TMs and even TONs, they're super fast, powerful method for quickly finding these thermal stable candidates, but thermal stability is not the only type of stability. You've got chemical and colloidal stability to think about as well, plus the overall activity of your antibody.
Yeah, for sure. And that is what is so nice about our Prometheus Panta, is that the colloidal stability from light scattering measurements can be measured in parallel with your thermal unfolding profile using nanoDSF. And this really informs independently on purity and the size of biologic, which you can actually follow the change in the size in response to that temperature gradient. And also the independence of these parameters is incredibly powerful. So we know that the most thermally stable candidate in the world is going to be no good and of no use to us if it is highly prone to oxidation or if it's just not active at all.
Yeah, thanks. That is definitely super critical to think about. And independent stability parameters are so critical for getting the most out of your developability work. And anybody watching can check out the link I mentioned earlier from our colleague Nathan if you'd like to learn even more about that data that we just showed and really deep dive into it. Okay, with that, we're a bit over and we have covered so much information about different experiments in biologics. So Bridget, I just want to give you the floor one more time. Do you have any parting thoughts for us before we sign off?
So yeah, just a couple. It will come down to careful planning with these experiments in order to get good quality data. And also, if you consider that poor sample handling will just confuse your data interpretation down the line.
Awesome. So Bridget, again, thank you so much for joining me to address all of these practical concerns around experimental design and data interpretation.